Yeah, we're going to continue moving on to save time for the panel for questions. So next is Quan Liu from Harvard University, who will be talking to us about identifying suppressors of stress response.
Good afternoon. So today I'm going to tell you a story that's currently under review, that's not published, but I hope that can help us illustrate how we can use CRISPR genome editing tools to help us understand the mechanisms of toxicity. In this case, the one environment toxin, the arsenic. In fact, the approach I'm going to describe actually is very similar to what Chris just described. So actually, you make my work much easier. So hopefully, this some of the stuff I'm going to describe will be easier to understand. So just to say that as an environmental scientist, I guess now I can call myself that. So arsenic is bad, and it's ubiquitous in the environment. Most people use this fact saying that more than 150 million people in the world are exposed to a high level of arsenic in the drinking water. But in fact, any level of arsenic is bad for us. So there's no safe level. Chronic exposure to arsenic has been associated with many diseases, including skin lesion, all kinds of cancer, skin cancer, black cancer, even lung cancer, neural problems, cardiovascular diseases, type 2 diabetes. So all this is why arsenic can have this multi-organ, dysregulation in the for human health. So as Chris already alluded to, actually there are many still controversy. What actually caused arsenic to have all this bad problem in our organs. So one potential mechanism for arsenic to have this very pleiotropic effect is that it's a Proteo-Toxicant. Essentially, it leads to protein misfolding. So what through this is that arsenic actually can interact with sial groups of a protein. Therefore, as you can imagine, then when you disrupt these this bond sial groups, that can lead to the protein misfolding. When protein misfold in the cells need to respond. So many, as you know, the ER, which is endoplasmic reticulum, is an organelle in a cell that responsible for synthesis of many of the proteins, including the protein gets secreted, the plasma membrane proteins. So here that in the ER, the endoplasmic reticulum, so how the ER respond to misfolded protein. So here is the pathway actually being mapped out, very well mapped out from yeast to human. It's ultra conserved. So essentially it's a three component pathway. Let me see if where is it here. So there are three components of sitting. This is the ER membrane. Sorry, this is hard to say. This is the nuclear membrane. Sorry, this is the ER membrane. So there are three protein components sitting on the ER. Actually, I'm not going to go into detail how, but essentially these are the sensors at the ER membrane. So anything that gets misfolded here can activate, tear this. This is IRE1. This is a PERK kinase, ATF4. So tear this. There's a problem in the ER. There's protein misfolding, and then send signal. I'm not going into detail how this signal works, but essentially in the end, go to the nucleus, activate these transcription factors that converge on one major pathway, this CHOP, which is master transcription factors that is responsible for the cells to decide whether they are going to commit suicide or not. So it's already known in the literature that many environmental exposures, such as arsenic exposure, can activate CHOP. It's well known, but how it does that is still not well mapped out. So here that we use genome-wide CRISPR screen that Chris described actually to help us understand the molecular mechanisms by which arsenic exposure activate CHOP, which is a central transcription factor responsible for decide whether the cells to undergo apoptosis. So how we do that? So the CHOP is a controlled transcription itself. So we basically took the CHOP motor, hooked up with M-cherry, which is fluorescent protein. So the idea is that if you don't have any protein misfolding, in this case, no arsenic, therefore the CHOP promoter is not active, therefore you don't have cherry expression. The cells is dark, you don't see anything. But if you expose the cells to the arsenic, now you activate CHOP. Now you have M-cherry expression, so cell become red. So as you can see here, this is the M-cherry expression as fluorescence. This is event of cell number. As you can see, increase amount of arsenic will increase the M-cherry expression. So this is a cell model that we are going to use to monitor the cell's response to arsenic. So here, because again, because Chris already did a very nice job describing how the genome-wide CRISPR knockout works. So basically, we took the genome-wide CRISPR knockout library, which is a lentivirus, and infect our cells that express this CHOP M-cherry, as I just described in the last slide. So essentially, if you end up with a library of cells, essentially it's a mutant cells. Each one of them contain one gene, individual gene being inactivated. So you have in a million cells, each of them contain one gene. So basically, this is a collection of mutant cells. So then we treat these cells with arsenic. So normally, as I described to you, that they will respond because it will activate the CHOP in promoter. Therefore, you have M-cherry expression. So idea is that if you hit a gene, you knock out a gene that's essential for the cell to respond to arsenic, you don't see M-cherry. So these cells with reduced or no M-cherry expression. If you hit a gene that's normally suppress cells respond to arsenic, now you remove that in suppressor, the cells will respond more dramatically than normal. Therefore, this is a redder, I would call it, redder in cells. By doing the flow cytometry or FACS, in fluorescence activated cell sorting, you can sort these different populations. And then in some cases, you need to resort them because just to make sure that they enrich this in extreme population to remove the bystanders. So using this approach, so here I don't have to go through these. People have really give a very nice introduction to CRISPR. So here is a library we use also. Chris lab uses this is a Gecko library from Fan Zhang's lab. Here I want to actually emphasize one thing. This was published in 2014, right after the publication of their paper. They deposited the whole library at Adigene for a fraction of cost. So basically, you can buy the whole library for $650. That's not a lot of money. So I think this is some people are asking this question about the availability of these reagents. Actually, the CRISPR field in general has been very open. Almost anything right after publication, you can get it. Same as the base editors that developed by David Liu's lab. Now you can buy it $60. So it's really amazing. So the other thing I want to emphasize about this one, actually, this library contains both the gRNA, the guide RNAs, the targeted genes for the protein coding, as well as for the targeting for some of the RNAs, particularly the microarray. So this will become relevant, as you will see. We identify both protein coding genes as well as microarrays that can regulate cells' response to arsenic. So here is how we do it. I'm just go briefly. You have library. You treat with arsenic. You see different response. In this case, we isolate the suppressors, which you remove a suppressor, the cells respond more. So basically, this is population cells. And then we do actually 3 rounds of sorting. And then in the end, we do deep sequencing on the barcode, as Chris mentioned. All these guide RNAs, they contain barcodes. So basically, you just PCR of them and then sequence them and compare to the unsorted cells. So here is what we did. I'm not going into detail. I think somebody asked a question about the high throughput nature of such screen. In fact, without in my collaborator, I wouldn't be able to tell you what I found. There's just AGCTs, But she was able to actually help us, make sense of the data. But the point of this analysis is that essentially you compare the sorted library, sorted the population with the unsorted population. So just see which one gets enriched, which one gets dropped out. And so here we just essentially convert the sequence into the fold of enrichment in the sorted sample. As Chris also mentioned, in this library, the built-in for this library, there are 6 guide RNAs per gene, 4 guide RNAs per microRNA. So therefore, you look for the number of independent guide RNAs in each of the analysis, because multiple independent guide RNA will increase confidence for that hit. So here is how we organize data. So basically, this is a fold change of each of the guide RNA. So sorry, each dot is 1 gene, the average of 6 guide RNAs. And then this is a fold of change. So how much they got enriched. And this is a number of guide RNAs. As you can see here, most of the stuff are here, just like they don't change much. They only 1 single guide RNA. These are the one you will ignore. Okay? So the one you need to focus is that there are multiple number of sgRNAs, as well as they have a higher enrichment fold. So those are the things we focus. So this we call a hit. From this one, I'm not going into detail. Among the top hits, we found there are 2 genes actually turned out in the same pathway that have been published actually. The one is called R3-MBTR2. Without the paper, I wouldn't go into this, because what the heck is this? Like, I don't even know what, I still can't tell you what this R3-MBTR2 stands for. But MGA, this is a max interaction protein. So anyway, those proteins actually are the components of this same pathway that's involved in the epigenetic regulation, gene expression regulation. In fact, we also identified the E2F6, but it's lower on our list of hits. So this gives us confidence. This is might be something interesting. So first, you need to validate this. So essentially, what we how do I go back? Okay, so we use this T7U1 assay. Essentially, this assay tells you whether there's a mutation, there's a insertion or deletion. So here, if you do that, you can see that different guide RNAs actually lead to the cleavage of this, indicating there's a mismatch, meaning that the CRISPR happens. This is a very quick, easy way to tell you that the CRISPR worked. Later on, we got antibody. We also show that the gene is knocked out. So you can do that, as you can see. So essentially, this is a reconstitution. You take the original chopped mCherry cells, remake these knockouts, and see whether you can get the phenotype. Essentially, yes, the cells become more sensitive, meaning that there's a higher expression of mCherry in the knockout cells in both the R3, MBTR2, and the MGA here. So this is and we also were able to demonstrate that these cells become more susceptible to cell death. So in this MGA, as well as R3, MBTR2, as you can see here, there's a number of cells, surviving cells. If you knock this out, there's less surviving cells. And this is into the mechanism, because this is not very relevant to this audience, to CRISPR. So I'm going to just summarize that we found out that using the chromatin IP assay, we were able to demonstrate this complex R3, MBTR2, and MGA, actually they bind to the promoter of CHOP. And normally, under a basal condition, when you have the ER stress, in this case, the arsenic exposure, this complex, suppressed complex, gets kicked out and then replaced with this more active complex. So that's how the CHOP expression happened here. So this is just show that, for example, this toxicant, which is another ER stress, and you can see here, this is remove the R3-MBTR2 from the promoter region. As I mentioned, the library also contains microRNAs. So we also actually identify, again, the same type of analysis, gene fold change, number of microRNAs. So we focus on this microRNA, including here. So this just show that, again, reconstitution puts the guide RNA of this microRNA back to the original cells and see whether they did anything. So as you can see here, that this is a control. You add arsenic, there's more immature expression, and this different sgRNA knockout. So knockout this miR-124, as you can see here, didn't change much at the basal level, but then when you add arsenic, the immature expression increased. Same for the other 184. Actually, we have in a bunch of others, but also that the point is that the one guide RNA did the same, the other, so it's very similar. So there are multiple guide RNAs for each one of the microRNA hits. So this is a mechanism, and we figure out actually it's through both by informatic analysis and also experimental analysis by transfecting this microRNA into the cells to identify which target of this microRNA actually regulates. So I'm not going to get into detail how microRNA works, but Dana actually gave a very interesting, very good primer on how some of these small RNAs regulate gene expression. So here, what we show is that we went on into the cells which have this microRNA there, and then we want to see which specific branches of the ER stress pathway is being perturbed. We were able to identify the IRE1, XBP1 branch. Remember, there are 3 branches of the ER stress response, and we show that it only affected the IRE1 pathway here. So, and this just show that, in fact, the microRNA 124 directly target the IRE1, which is in canonical central pathway of the ER stress. So, for example, if you transfect this microRNA mimic into the cells, you can see it suppresses the expression of the IRE1. In this case, we use luciferase as a reporter. And if you mutate the targeting sequence of this gene in the 3' UTR, 3' untranslated region of the IRE1, you essentially eliminate this suppression by the microRNA 124. So this, just to summarize that, through the screen, we identify many suppressors of ER stress response in response to arsenic exposure. We characterize the mechanism of, detailed mechanism of 2 hits. One is this microRNA 124, the other is L3-MBTL2 protein complex. So to summarize that, using a cell-based assay that we can monitor the cells response to arsenic, we did the genome-wide CRISPR screen. We identified both protein coding genes that can regulate cells response to the arsenic. We also identify microRNAs that can regulate the response. One thing I didn't have time to talk about is that, in fact, I think people mentioned about this reverse genetics. I think in this case, we use a sort of a forward genetics to identify the genes that are important for a particular phenotype, which very relevant for the health impact of the arsenic exposure. But then we can go back actually into the humans and ask whether there are genetic variants in the genes we identify that may alter the phenotype in humans. So in this case, actually, we were able to identify several SNPs, which is a small genetic variation in the microRNA 124 gene that are strongly associated with neurodevelopment, in particular in the context of arsenic exposure in a human cohort. So I think this is probably a very exciting direction that we are very interested in to follow. So finally, thanks to people who did the work, people in my lab and my collaborator for this particular project. As Chris mentioned, that we also have a tox in CRISPR project that is not described here. So I will stop here. Thank you.